Hello and good day everybody. Hope all of you are doing fine. Now for this lesson, we shall study the physiology of a neuron or how nerve impulses are transmitted. Now when we speak of nerve impulse or action potential, this refers to the electrical current moving from the dendrites to the cell body to the axon of a neuron. Neurons are the unit structure of our nervous system. And when we speak of nerve impulse or action potential, this is the result from the movement of ions or electrically charged particles into and out of a neuron through the plasma or cell membrane. Now let's have a quick review of the different parts of a neuron with their respective functions. So we have here the cell body. The cell body is surrounded by those branching structures called dendrites. These dendrites, they receive signals from other cells. Then attached to these dendrites is the cell body or soma that organizes and keeps the cell functional with its nucleus that controls the entire neuron. And attached to the cell body or soma is the axon that transfers signals to other cells and organs. The axon is covered by a myelin sheet that increases the speed of the signal. And the myelin sheet is produced by the Schwann cell. The Schwann cell produces the myelin sheet which acts as an insulator. Then in between Schwann cells is the node of Ranvier that allows diffusion of ions. Then at the end of the axon is the axon terminal that forms junctions with other cells. Now, as I mentioned earlier, the transmission of nerve impulses or action potential is the result of the movement of ions or electrically charged particles into and out of the neuron through the plasma or cell membrane. Now these ions, they move into and out of the cell through special channels called ion channels embedded in the cell or plasma membrane. These are special proteins in the plasma or cell membrane that allow specific ions to pass through. Some of these ion channels remain open all the time. Others will open and close depending on the conditions of the cell and its environment. Now there are two general types of these ion channels. We have the so-called ligand-gated ion channel and voltage-gated ion channel. For ligand-gated ion channel, there is a substance, usually a neurotransmitter, that binds with the ion channel. And as the neurotransmitter or the ligand binds with the ion channel then opens, allowing ions to pass through. Now for voltage-gated ion channels, these channels will open depending on the membrane potential or the voltage. So I hope everything is clear about the different types of ion channels. Again, we have the ligand-gated ion channels in which ligand or neurotransmitters are involved. These neurotransmitters binds with the ion channel, eventually opening the channel. Then we have the voltage-gated ion channel that opens the channel depending on the membrane potential or the voltage. So at this point, let us now study the actual transmission of nerve impulses. Now, before a neuron can transmit nerve impulses, it must be in a so-called resting membrane potential state. Now, what is this resting membrane potential? Now, resting membrane potential means that the neuron is inactive. The neuron is not receiving any stimulus at all. And there is an imbalance between positive and negative charges across its membrane. So this difference in charges will result in a potential called the resting membrane potential. Now, in order to create this so-called resting membrane potential, the sodium-potassium pumps, as we mentioned earlier in my previous lecture about the activities of the cell membrane, must transfer sodium ions out of the cell and potassium ions in the cell with a 3 to 2 ratio. So as we have learned before in that lesson on the activities of the cell membrane, the sodium-potassium pump pumps 3 sodium ions out and 2 potassium ions in. This will create a charge imbalance across the membrane of approximately negative 70 millivolts. So here is an animated illustration to help you visualize what's going on in the creation of a so-called resting membrane potential. So we have here the sodium-potassium pump. So these are special channels that pump sodium ions out of the cell and potassium ions in. Specifically, we have here 3 sodium ions that are pumped outside the cell and 2 potassium ions enters the cell, creating a difference in the concentration of sodium and potassium ions in and out of the cell. So if the process continues, there will be more sodium ions outside the cell compared to the amount of sodium ions inside. So again, in this sodium-potassium pump, 3 sodium ions exits the cell and 2 potassium ions enters the cell. Alright, so that's the sodium-potassium pump. And this sodium-potassium pump will create a difference in charges in and out of the cell. So eventually, if the process continues, the outside of the cell becomes positively charged and the inside of the cell becomes negatively charged. So this is what we call a resting membrane potential. And again, why is there a difference in charges? Again, there is 30 times more potassium ions inside the cell than outside, and about 15 times more sodium ions outside than inside. Again, remember, in the sodium-potassium pump, 3 sodium ions exits the cell and 2 potassium ions enters the cell. And there are also large negatively charged proteins trapped inside the cell. That's why the inside of the cell is negatively charged and outside is positively charged. So this is what happens when the neuron is in a so-called resting membrane potential. So would you expect to see so much potassium inside and so much sodium outside the cell? So special protein channels, again called the sodium-potassium pump, move 3 sodium ions out and bringing 2 potassium ions back in when the cell is at rest. In a resting cell, there are no open channels for sodium ions to easily move back into the cell. However, there are some potassium channels open all the time. Sodium ions causes the outside to be positive, forcing more potassium ions into the cell. So this is what we call a resting membrane potential. So I hope everything is clear about the role of the sodium-potassium pump in creating a so-called resting membrane potential. So again, the sodium-potassium pump pumps 3 sodium ions out of the cell and allows the entry of 2 potassium ions. So this will create a difference in the charges. So outside of the membrane, it becomes positively charged, and inside the membrane, we have a negatively charged area. So this difference in charges will create a so-called resting membrane potential of the neuron. So in a resting membrane potential again, the membrane is positively charged outside and negatively charged inside. Alright, so before we move on, let's have a quick review of what we have learned so far. Alright, so let's fill up the following. So in a resting neuron, the inside of the cell has a blank charge. So what do you think is the answer? So if you think that the answer here is negative, you got it. So in a resting neuron, the inside of the cell has a negative charge. Alright, what about the second statement? In a resting neuron, potassium ions are high in concentration blank of the cell. So what do you think? If your answer is inside the cell, then you got it. So in a resting neuron, potassium ions are high in concentration inside of the cell. Remember the sodium-potassium pump. So the sodium-potassium pump pumps 3 sodium ions out and 2 potassium ions in. Eventually, if the process continues, there will be a high concentration of potassium ions inside the cell. Alright, and for the last statement, the resting membrane potential of a neuron is about blank millivolt. So if your answer is negative 70 to negative 90 millivolts, then you got it. So at this point, let us now study how a nerve impulse is generated. So a nerve impulse is generated once the neuron receives a stimulus. So the neuron gets stimulated, either receiving a signal from another neuron. This stimulation of the neuron causes a change in the resting membrane potential. If a neuron is stimulated enough, the inside of the cell will reach a critical level called a threshold, about negative 55 millivolts. And at this point, upon reaching the threshold, the sodium ion channels will open. And as the sodium ion channels will open, this will cause a change in the resting membrane potential, which we call a depolarization of the neuron. So at this point, let us now study this next step. Once the neuron gets stimulated, the neuron now is depolarized. Now, what do you think will happen now once the neuron gets stimulated? Now, depolarization will occur. Now, in depolarization, sodium ions, once the neuron gets stimulated, rush into the neuron because of diffusion forces, wherein the sodium ions will move from a region of high to low concentration. And once the neuron gets stimulated, it will open the sodium ion channels once the threshold is reached. The charge now inside the cell will eventually reach about positive 30 millivolts. Relative to the outside of the cell, the inside now becomes positively charged and the outside becomes negatively charged. And at this point, the sodium ion channels will close. So this change in polarization, wherein the inside of the cell becomes positively charged and the outside of the cell becomes negatively charged, is called depolarization. So remember, in resting membrane potential, the outside of the cell is positively charged while the inside of the cell is negatively charged. Now, once the neuron gets stimulated and upon reaching the so-called threshold, the sodium ion channels opens up, allowing the entry of the sodium ions, and this will now create a change in polarity. So the inside of the cell now becomes positively charged and the outside becomes negatively charged. So this is what we call depolarization. Alright, so again, in depolarization, you have here a stimulus. So the neuron gets stimulated and the membrane gets depolarized. A depolarized membrane allows the entry of sodium ions, again because of the threshold, the sodium ion channels opens up, allowing the entry of the sodium ions inside the membrane. So eventually, the inside of the membrane becomes positively charged and the outside becomes negatively charged. This exchange of ions initiates an action potential in the neuron. So let's take a look at this animated illustration showing the process of depolarization. So once the neuron gets stimulated, the sodium ion channels opens up, allowing the entry of the sodium ions. And this entry of the sodium ions will create now a change in the charges wherein the inside of the cell becomes positively charged and the outside of the cell becomes negatively charged. So there's now a change in polarity, and this is what we call depolarization, and this drives the transmission of the nerve impulse. Now the next step after depolarization of the membrane will proceed to repolarization. Now what is repolarization? So in repolarization, we have now the opening of the potassium ion channel. So the potassium ion channels open up and this causes the potassium ions to rush outside of the cell. And as the potassium ions leaves, it causes the inside of the cell to become negative again, about negative 70 millivolts. And this is referred to as repolarization. So again, we go back to the resting membrane potential state. So let's take a look at this animated illustration showing both processes of depolarization and repolarization of the cell membrane of the neuron. So once the neuron gets stimulated, the sodium ion channels opens up and this will allow the entry of sodium ions. Once the sodium ions enters the cell, there will be a change in charges wherein the inside of the cell becomes positively charged, then the outside becomes negatively charged. So there is a change in polarity, and that's what we call depolarization. Then after depolarization, the membrane now goes to the so-called repolarization step, wherein this time the potassium ion channels opens up, allowing the exit of potassium ions. And this will create a change in charges again. So inside the cell, you go back to being negatively charged and the outside becomes positively charged. And eventually, after repolarization, the cell membrane returns to its resting membrane potential state, wherein again outside is positively charged and inside is negatively charged. And if the process continues, the polarization and repolarization of the membrane, the nerve impulse is transmitted from dendrites to cell body to the entire length of the axon. So isn't that fascinating indeed? Now before we proceed, let's watch this video to further explain the transmission of nerve impulses. Alright, let's watch this.
This is a neuron, which has four main parts. The dendrites receive information, the cell body processes and integrates that information, the axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long as they often need to transmit information over long distances. As we just saw, the dendrites are the part of the neuron that receives incoming signals. Based on the strength of this incoming stimulation, the neuron must decide whether to pass that signal along or not. If the stimulation is strong enough, the signal is transmitted along the entire length of the axon in a phenomenon called an action potential. When this happens, we say the neuron fires. Transmission of a neuronal signal is entirely dependent on the movement of ions or charged particles. Various ions, including sodium, potassium, and chloride, are unequally distributed between the inside and the outside of the cell. The presence and movement of these ions is not only important when a neuron fires, but also at rest. To start, let's think about the positively charged sodium and potassium ions. When a neuron is not sending a signal, it is considered to be at rest. In a typical neuron, in its resting state, the concentration of sodium ions is higher outside the cell than inside. The relative concentration of potassium ions is the opposite, with more ions inside the cell than outside. This ionic separation occurs right at the cell membrane and creates a chemical gradient across the membrane. Because ions are charged particles, we also need to consider their charge when thinking about their distribution across the membrane. At rest, there are more positively charged ions outside the cell relative to the inside. This creates a difference in charge across the membrane, which is called an electrical gradient. Together with the chemical gradient we already mentioned, we refer to this ionic imbalance as the electrochemical gradient. The difference in total charge inside and outside of the cell is called the membrane potential. At rest, when no signals are being transmitted, a neuronal membrane has a resting potential of approximately -70 millivolts. This means that the inside of the cell is approximately 70 millivolts less positive than the outside. Both the chemical and electrical gradients we just discussed contribute to establishing this potential. While the inside of the cell has a net negative charge and the outside of the cell has a net positive charge, the charges line up at the membrane and the bulk solution on either side is actually electrically neutral. The resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means that the concentration gradient and the electro gradient for each ion is equal and opposite. Ions cannot simply move across the membrane at will. Instead, they need a protein embedded in the membrane to facilitate their movement. Most ions cross the membrane through a structure called an ion channel. Ions move through channels by passive diffusion along their concentration gradient. Some ion channels are always open, but many require a signal to tell them to open or close. For example, voltage-gated channels only open when the membrane potential reaches a certain value. On the other hand, ligand-gated ion channels are triggered to open when they are bound by a specific molecule. Mechanically gated ion channels open in response to physical forces such as changes in length or changes in pressure. Most ion channels are selectively permeable, meaning that they only allow one or a small subset of ions to pass through. Voltage-gated ion channels, for example, typically only allow a single ion to cross the membrane when they open. This means that we need separate channels for each ion, i.e., voltage-gated sodium channels, as well as voltage-gated potassium channels. As ions move through a channel and cross from one side of the cell membrane to the other, they cause the membrane potential of the cell to move away from its resting potential. If the resulting change in membrane potential is small, we call this a graded potential. Graded potentials can vary in size, can be either positive or negative, are transient, and typically do not result from the opening of voltage-gated ion channels. When ion channels open and a graded potential occurs, the neuron moves quickly to reset its membrane potential to resting values. This is accomplished primarily by the use of the sodium-potassium pump, which uses the energy generated by ATP hydrolysis to actively transport ions across the membrane against their concentration gradient. In other words, sodium is transported to the outside of the cell, where its concentration is higher, and potassium is transported back into the cell, where its concentration is higher. One cycle of this pump transports 3 sodium ions outside the cell and brings 2 potassium ions inside the cell. This unbalanced charge transfer contributes to the separation of charge across the membrane and also to the ionic concentrations we see at rest, thus restoring the chemical and electrical gradients to their resting levels. Maintaining these ionic balance in neurons is so important that this process can account for 20 to 40% of the brain's total energy use. Only when the resting membrane potential and ion distributions are maintained at precise levels will the neuron be poised and ready to fire an action potential. When the outside stimulation is large enough to bring the membrane potential in the neuron body up from -70 millivolts to the threshold voltage of -55 millivolts or higher, this triggers an action potential at the axon hillock, which then travels down the axon. Voltage-gated sodium channels have 3 states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position and sodium rushes into the cell because of the electrochemical gradient. As positive sodium ions enter the cell, the membrane potential becomes less negative and more positive as it approaches 0 millivolts. This is called depolarization. Eventually, the voltage gradient goes to 0 and beyond 0, up to a positive 30 millivolts. This is called an overshoot. As the membrane potential becomes positive, the sodium channel inactivation gate shuts, making the channel inactivated. This stops the flow of sodium ions into the cell. The change in membrane potential also opens the voltage-gated potassium channels, though they open and close more slowly. Because of the potassium electrochemical gradient, potassium ions flow out of the cell, making it less positive and eventually negative. This process is called repolarization. Because the potassium channels are a little slow to close, for a brief period the membrane potential is hyperpolarized. It's more negative than the resting potential. During hyperpolarization, the potassium channels close. Throughout all this, the sodium-potassium pump is still working. The pump restores the chemical gradients by putting the sodium and potassium back in place. And the pump reestablishes the potential gradient by moving more sodium ions out than potassium ions in. This returns the membrane potential back to its resting potential. During repolarization, the inactivated sodium channels won't respond to any stimulus at all. During this time, the neuron is in its absolute refractory period, the period of time when a nerve cannot fire another action potential no matter how strongly it's stimulated. The absolute refractory period prevents action potentials from happening again too quickly and prevents the action potential from traveling backwards along the axon. During hyperpolarization, the sodium channels are closed and the inactivation gate opens. There is no change in sodium flow, but now they could be opened again. This is called the relative refractory period, because while the sodium channels could open, it would take a larger than usual stimulus to reach threshold. because the cell is hyperpolarized due to the potassium still leaving the cell. The amplitude of the action potential for a particular neuron, that is, the maximum voltage in one neuron during an action potential, never changes. An action potential doesn't get bigger with a bigger stimulus. It's all or nothing. It either happens or it doesn't happen. What can change is the frequency of the action potential. A neuron might fire many more times per second in response to, say, an intense pain, and less frequently in response to a gentle breeze. Some axons transmit action potentials faster than others. One variable that increases conduction velocity is the presence of myelin sheaths around axons. Myelin speeds up transmission through a process called saltatory conduction, in which the action potential signal appears to jump along the part of the axon covered by the sheath. In the peripheral nervous system, the sheaths are formed from glial cells known as Schwann cells. There are small gaps between Schwann cells called the nodes of Ranvier. The action potential appears to jump from node to node, speeding the transmission. In the central nervous system, the sheaths are made by cells known as oligodendrocytes. To review, with no stimulus, the membrane is at its resting potential. A small stimulus causes a graded potential, and a stimulus above the threshold creates an action potential and the neuron fires.